All right, today we're diving way down to the microscopic level. We're going to crack open the survival guide for one of the most successful and invisible life forms on our planet, bacteria.
Okay, let's just start with this number, 1,048,576. What on earth is that? Well, get this, that's how many bacteria can come from a single cell in under 7 hours. Yeah, from 1 to over a million, just like that.
So that brings up the big question, right? How is that even possible? I mean, it sounds like pure chaos, but it's not. It turns out this explosive growth actually follows a very strict, very organized set of rules.
So, what are these rules? Well, the first thing you have to understand is that for bacteria to grow like this, everything has to be just right. You can think of it kind of like a recipe, and that recipe has two main parts.
On one side, you've got the physical stuff. We're talking about the environment's temperature, its pH level, how acidic it is, and this thing called osmotic pressure. Then, on the other side, you have the chemical ingredients. These are the literal building blocks they need to make more themselves. Things like carbon, nitrogen, and for some, oxygen. So let's figure out what it takes to build the perfect bacterial home.
So, what does this perfect home even look like? Well, as it turns out, bacteria can be surprisingly picky about where they live. Let's start with temperature. For bacteria, it's all about that Goldilocks zone. Not too hot, not too cold. Every type has its own perfect sweet spot. If it gets too cold, they slow down. Too hot, they stop completely. You can see it right here in this graph. You've got your cold lovers, the psychrophiles, over on the left. On the right, you have the heat lovers, the thermophiles. And right there in the middle, those are the mesophiles. A lot of the germs that make us sick fall into this group because, well, they just love our cozy body temperature.
And it's pretty much the same deal with pH. Most bacteria are really particular. They like things to be neutral, right around a pH of 7. Very few can survive in something super acidic or super alkaline. And that's exactly why we use things like vinegar for pickling. It makes the environment way too acidic for them to handle.
Okay, next up is something called osmotic pressure. It's really all about water balance. So think about it like this. Water naturally moves from a less salty place to a much saltier place. Bacteria are mostly made of water, so what happens if you drop one into, say, a super salty or shootery solution? All the water inside that cell just rushes out. The poor thing literally shrivels up. This process is called plasmolysis, and it's the whole secret behind why salting meat or making jams has been a great way to preserve food for centuries.
Now, what about oxygen? For us, it's simple. We need it to live. For bacteria? Oh, it's complicated. Check out these test tubes. On the far left, you see the obligate aerobes. They're just like us. They need oxygen, so they all crowd around the top. But then look at the next tube, the obligate anaerobes. To them, oxygen is pure poison, so they're hiding way down at the bottom. And then you've got the really adaptable ones in the middle, the facultative anaerobes, who are basically like, oxygen, no oxygen, I can make it work. This incredible diversity is a huge reason why you can find bacteria pretty much anywhere on Earth.
Okay, so let's imagine we've set the stage. The conditions are absolutely perfect. What happens now? Well, we are about to watch the entire dramatic life story of a bacterial population unfold. And trust me, it's a drama in 4 acts.
And this curve right here, this is their entire life story all mapped out. It shows you the number of living cells over a period of time, and it almost always follows these same 4 distinct phases. So let's break it down phase by phase, starting from the beginning. Act 1 is the lag phase. Now, looking at the graph, it seems like nothing is happening at all, right? The line is just flat. But that is totally misleading. Under the surface, these cells are going crazy. They're making all the tools and enzymes they need. Basically, they're gearing up for what's about to come.
And then, bam, Act 2, the log phase. This is the population explosion. The numbers just skyrocket. They're doubling and doubling again at a crazy fast rate. This is where that million-plus number we talked about becomes a reality. They are just reproducing as fast as they physically can.
But, no party can last forever. We hit Act 3, the stationary phase. The growth screeches to a halt. Why? Well, they're starting to run out of food, and their own waste products are building up. So for every new cell that's made, another one dies off. The population just hits a plateau.
And that leads us to the final act, the death phase. The party is officially over. The environment has just gotten too toxic and there's not enough food to go around.
Now, way more cells are dying than are being created, and the population starts to decline, steadily, until it might disappear completely.
So we have this nice, predictable curve. But hang on a second. If these organisms are totally invisible, how in the world do scientists actually count them to even make a graph like this in the first place?
Well, one of the most common ways is a technique called serial dilution. The logic is actually pretty simple. You can't count a billion of anything, but you could probably count, say, 50 or 100. So scientists take the original sample and just keep diluting it by 10 times, then 100 times, 1,000 times, until they get a sample that's watered down enough to grow a manageable number of bacteria.
Once they have those dilutions, they need to grow them on a petri dish, and there are two main ways to do it. You've got the pour plate method, where you actually mix the sample right into the jelly-like agar. Then there's the spread plate method, where you just spread it right on top. But either way, the goal is exactly the same. Get individual bacteria separated from each other so that each one can grow into its own visible dot or colony that you can actually see and count.
But here's the thing. Everything we've talked about so far is basically bacteria floating around by themselves in a lab. Out in the real world, survival isn't a solo mission. It is all about community. And when it comes to bacterial communities, the ultimate example is something called a biofilm. And no, this isn't just some random layer of slime. It is a highly organized, cooperative society of microbes. They literally build their own protective home out of this sticky stuff they make themselves. You should really think of it as a full-blown bacterial city.
And the way these cities are built is just incredible. Look at how they form these pillar-like towers. The spaces in between aren't just empty space, they're actually channels. It works like a primitive circulatory system, letting water flow through to deliver food and wash away waste. For tiny microbes, this is a seriously sophisticated survival strategy.
Now, living together like this has massive benefits for the bacteria. They share food, they protect each other from danger. We actually even harness this power in things like sewage treatment. But there's a major downside for us. Biofilms are the gunk that clogs your pipes, they cause really stubborn infections on things like medical implants. And here's the really critical part. Bacteria in a biofilm can be up to 1,000 times more resistant to antibiotics than the free-floating kind. That makes them a huge, huge challenge in medicine. So when you look at the whole picture, from the precise rules that govern their individual growth to the complex power of community, it's clear that bacteria have truly mastered the art of survival. Their story is a powerful reminder that even the tiniest organisms can achieve incredible things through adaptation and cooperation. And that kind of leaves us with a final thought, doesn't it? What can we, in our much larger, more complex world, learn from their ancient survival playbook?